Today, I'm going to do a short lecture on the scientific method. So on my first screen here, it shows a diagram with the steps of the scientific method. But one thing I want to stress here is that this is not written in stone. This is not a series of steps that we absolutely do in order as researchers, right? Sometimes we skip around a little bit. So this is more of just a guideline on the scientific process. But let's talk about some of the steps here. So first we have making observations. And this step occurs sometimes, and let me explain that. So I could be walking out in nature, right? I think I mentioned before that I study pollinators. So maybe I'm walking out in a grassland and I observe something that I think is unusual, right? Like maybe I see male bees sleeping in certain types of flowers, which actually happens, by the way, it's kind of cool. So I might develop a research question based on that. Like, do they sleep in all types of flowers or just this certain type of flower, right? So in that case, an observation did give me my start on this particular research question. But other times, and this is what usually happens, as scientists, we read a lot of scientific papers that have to do with whatever it is we're studying, right? So many times I have read a paper and I've thought of extensions of whatever study they were doing. Like, oh, that's an interesting study, but I wonder what happens if this occurs. And so I start thinking about questions as I read the paper. And then I do background research to see, has anybody answered that question, right? So in that case, I am starting with thinking up interesting questions. Okay. Now, the second or the third step actually is to formulate a hypothesis. Now, most of you have probably heard the definition of a hypothesis as being an educated guess. But I am going to change that to a different definition. So a hypothesis is a probable answer to your question based on prior knowledge and background research. Okay, a hypothesis is a probable answer to your research question. Now I'm going to apply this to an example in the next few slides. Now, from your hypothesis, you should be able to develop a prediction. Okay. So the prediction is what you expect to observe given your hypothesis in different situations. Okay, what you expect to observe given your hypothesis when you look at different situations. Okay, so basically it's like if my hypothesis is supported, then I expect to see this. You're going to gather data and test. Now this says predictions. I'm actually going to change that to hypothesis. Okay, so you're gathering data to test your hypothesis. I'm going to create experiment to test your hypothesis. And this is where you collect your data. Now, after you have tested your hypothesis, you really move on to this step right here. You either support or reject your hypothesis. Okay, the hypothesis cannot be proven. And I want you to take a second and think about why that might be. Why can you not prove a hypothesis? Okay, I wrote here, a hypothesis cannot be proven, only supported. In order to prove something, you're implying that you have 100% certainty that your conclusion is correct, right? But there is no way you could test every possible situation. So we just say our hypothesis is supported. Now, this last part here, develop general theories. So I'm looking over here at this last circle. So a lot of people confuse theory with hypothesis. Okay, you'll hear people say like, oh, I have a theory about why that happened. Well, that's not a theory, that is a hypothesis. So a theory has been tested many times in different experiments and found to be supported, right? It explains many different observations. Okay. So a theory has been tested so many times, we're like 99% sure that it's correct. Okay. Like the theory of evolution. So now let's apply these steps of the scientific process. Now, let's say I am out bird watching and I notice a certain species of sparrow made all its nest with grass. Okay, well, that's an interesting observation. So my question could be, why are the nests of sparrows made with grasses rather than twigs? Right, because we commonly think of twigs in bird nests. So I want to know, why are sparrows making their nests out of grass instead of twigs? So here is my hypothesis. Remember, this is an explanation to your question or probable answer. So the hypothesis in this case would be sparrows use grasses in their nests rather than twigs because grasses are more abundant in their habitat. There's a lot more grass than twigs. And so I think that that is why sparrows use grass, because there's more available. So what I want you to notice about this hypothesis is, it uses a because statement to explain your answer to the research question. Okay, so you have your research question. I observe this. Why do I observe this? Hypothesis is your explanation. Okay, here is the because statement. I think this is occurring because grass is more abundant. Now, you won't always see like the because part, but if it's an explanation, it's a hypothesis. Now, down here is our prediction. If sparrows use grass because it is more abundant, and I compare areas where twigs are more abundant, the sparrows should make their nest with twigs. Okay, so the prediction is saying, okay, well, if this is my explanation is true, and I look at this new situation, I should see this. Here's what I expect. So the prediction is an if then statement. Okay, if my explanation is supported and I observe nest building in a new situation, then I expect to observe whatever it is you expect to observe. Whoops. Okay, so if my explanation is supported, that sparrows use grass because it is more abundant, and I observe nest building in a new situation, right? Now there's more twigs, then I expect to observe nests with twigs. Okay, so the hypothesis is your explanation. The prediction is what you expect to observe in certain situations. And a lot of times the prediction reflects what we're going to do in our experiment. Like this person, this researcher, would go to habitats with more sticks. That's her experiment. So this is just restating what I gave you, the difference between hypothesis and prediction. So it's important that you understand this. So a hypothesis is an answer and explanation to your research question. Whereas a prediction is the expected outcome in a certain situation. And I'm going to say the actual experiment, right? What you're actually doing in the experiment. So the prediction is like what you expect to see as an outcome of your experiment based on your hypothesis. Okay, it's based on your hypothesis. So let's move on here. Okay, there are two types of scientific studies, observational and experimental. So observational is what people do in health studies, but also what people like researchers like myself do in natural areas. Okay, so you take a small sample, you look at your results and you apply it to the population, right? But the important part of an observational study is you cannot control the assignment of your subjects to different groups. So let me write that down for you. Okay. So you cannot assign your experimental subjects, which could be like humans, animals, whatever, right? Just depends on what you're studying. Cannot assign your experimental subjects to different groups. And I'll explain what that means. I'm actually going to add a word here. Sorry about that. If you're writing this down, you cannot randomly assign your experimental subjects to different groups. So let's look at what that means. By the way, I'm going pretty fast as I write these things down. You just pause the video, right, to get it all. Okay, so let's look at this hypothetical study. We want to know if there is a link between having an abortion and getting breast cancer. Now, there's no judgment in this example, right? It's just a study that was done. So in order to say abortion causes breast cancer, we would have to do a controlled experiment, which is what I'm going to talk about next. So what I want you to note here is that observational studies can only establish correlation, not causation. So I could say it seems like breast cancer may be related to women getting abortion, right? But I can't say abortion causes breast cancer with an observational study. So let's look at this. In a controlled experiment, there would be a large group of pregnant women. I would randomly assign some pregnant women to get abortions, some to not get abortions. Then I would do cancer screening, years after the procedure for both groups, right? I'd cancer screen my women who had their babies, cancer screen the women who had abortions. Well, obviously this is not ethical. No pregnant woman is going to volunteer for this study, right? Sure, randomly assign me to get an abortion. So you have to do an observational study. Okay, you would have a bunch of women who already had abortions. And then you would do cancer screening and probably compare it to your general population. But the fact that you're not randomly assigning women, that you're taking women who already had an abortion, that makes it observational, right? You can't randomize the study and put some women in one group, some women in the other. You're already getting women with abortions, and there may be factors there that would control your results, but they're not the ones you're testing, right? If you get all women who've had an abortion, maybe, I don't know, maybe they're older, okay? And so maybe that fact that they're older and pregnant might affect breast cancer. You just don't know, okay? And I'm not saying this is true. I'm not saying it's older women who get abortions. I'm just trying to think of an example. So let's look at experimental studies. They are used to establish causation. And I put that right here for you. Determine cause and effect, same thing. And then you control variables. So let's apply this. So this is a weird study I found. I thought was kind of amusing. Some researchers wanted to know if dogs have a preference for music, like different music types, or if they have a preference at all, like does it affect their behavior? And like I said, this is an actual study. So they took a bunch of dogs, right, put them in a room, play music for them, see if it changes their behavior. Now, the first thing I want you to know about this is there are control variables that they need to pay attention to. So a control variable is a factor that may affect the results of the study, but are not but is not the factor being tested. Okay, so look here, I put some control variables in the example. These would be, pen's not working. Okay, it would be things like volume of the music, presence of humans, temperature. I would also think like time of day, maybe. So say you varied the volume for all the different dogs. How do you know it's not volume affecting their behavior rather than the music? Right? So you want to keep all those control variables the same between your groups. Okay. So to see if dogs change their behavior in response to different kinds of music, they split them into two groups, randomly assigned them to a group that receives the music treatment and a group that gets no treatment, no music. So they actually found out that dogs are calmer and more relaxed when listening to reggae. Kind of funny. I do find reggae relaxing. Okay, so let's break this study apart and learn about the parts of an experimental study design. So first of all, there are two groups, like I told you. The dogs are assigned to two different groups. One is the treatment group, one is the control. Actually, I need to write that. So the treatment group is the group that receives the treatment. Different types of music, right? The treatment group are the dogs listening to music. The control group is the dogs not listening to music. And that is so you can look at normal dog behavior with no music. How do you know if music changes their behavior if you don't know what their behavior is? Now, I want to make a distinction here. The control group is not the same as control variables. Okay, so the control group is the group of subjects that are in the normal or standard conditions. Okay, these are used for comparison. So the control group in this case is going to be dogs just, I guess, sitting in the room, but not listening to music. So now you know how sitting in the room affects dog behavior. Now, remember, that's the control group. Control variables are those factors that you keep the same between the groups. So let me write that actually. Whoops. Let me erase this. Okay, so control variables are the factors that are kept the same for both groups, like volume of music, etc. So you keep them the same for both groups, but you also keep them the same for each test, right? Each dog. Okay, hopefully that makes sense. So control variables are different than the control group. Okay, so you can see here, I wrote under control group, the dog's not exposed to music. Now, the other thing you need to pay attention to when you're thinking about an experimental study are your independent and dependent variables. So I wrote here that your independent variable is the variable that the researcher is manipulating or changing. It's the thing you're testing, right? whether a change in the independent variable results in a change in the outcome. So when you change music, what happens to behavior? That's the outcome. So I put as an example, the type of music in the dog experiment. And then you have the dependent variable, the outcome that is being measured. So in the example, behavior of the dogs. You're changing music, you want to look at behavior. Music is independent or treatment variable. Behavior is the dependent variable. It's the data you are collecting. So the final part of learning about the scientific method is to understand how to display your data. Okay, how to construct a graph and read a graph. You will have to do this later in the course. You're all going into the health professions, so I think it's really important that you understand how to read data. I mean, you may have to like read a scientific paper or something like that to learn more about your field. So it's good if you can kind of look at these graphs and decipher them. So few things you need to know. The independent variable, temperature, right? Oh, I'm sorry. So this is actually an experiment where it's based on this question: does temperature affect the number of times a cricket chirps in a 30-minute period? Okay, so the setup of the experiment, let me write that here, is crickets. I'm going to put a group of crickets are kept at, I can't remember, so 15 Celsius, I can't do the degree. I think I did 22 Celsius, 27 Celsius, and 33. I put Celsius because that's what we use in scientific papers. We never do Fahrenheit. So let's say in this experiment, I have my group of crickets. I keep them at 15 degrees Celsius. Okay, so at each temperature, cricket chirps are counted for 30 minutes. Okay, that's my setup for my experiment. So my independent variable is temperature. I'm putting them at different temperatures. I want to see if temperature affects cricket chirps. So my temperature always goes on the x-axis. Independent variable. The dependent variable is always put on the y-axis. Okay, so the number of cricket chirps. Now, you want to create axes with evenly distributed numbers. So like here, I increase by 5 every time. 10, 15, 20, right? I don't just put the temperatures on there, like have a line for 15, have a line for 22, right? I do even increments. Now here I wrote number of cricket chirps and they're even. They go up by 5, okay? So your axes, the numbers on them have to be evenly distributed. Now you also need to provide a title and axis labels so that the reader knows what they're looking at, right? So your title should be descriptive. Variation in cricket chirps versus temperature. And then my axis labels are temperature, and you have to put the units, degrees Celsius, and number of cricket chirps on the y-axis. Now, I want you to take a second and look at this graph and think in your head, what is this graph telling me? So the graph indicates that as temperature increases, so does the number of times the cricket chirps, crickets. Whoops, misspelled that. Okay, until 27 degrees Celsius, at which point cricket chirps decrease. Okay, so here is a positive correlation. As temperature increases, cricket chirps increase. Okay, they're both going up. This would be a negative correlation. As temperature increases, chirps go down. Okay, they're opposite directions. One's going up, one's going down. That's a negative correlation. Okay, so this concludes what you need to know about for the scientific method. If you have any questions, feel free to email. We can meet, we can email, whatever works best for you.